Welcome back to BioWorld. In my previous video, I shared with you the analysis based on topics. Today, I'm going to go into details of analysis based on subtopic to help you minimize your study load. So come join me. Here we have the analysis based on subtopic for chapter 12, where it is my opinion that 12.2 and 12.4 would be tested this year. I'll just share with you my simple logic for this. If you look at 12.2, in the year 2013 and its repeat, it was questioned back to back. 2018 and repeat, it was also questioned back to back. So I feel 2022 would be similar. And the reason why I'm choosing 12.4 is because I feel that it has been a very long time since it was tested. So let's have a look at what is in these two subtopics. For development of immunity, you need to know cell-mediated response and humoral immune response. Now remember, in cell-mediated response, we are talking about the role of T cells, and in humoral response, we will be talking about B cells. And naturally, when we talk about B cells, this will be related to the antibody reaction. Includes for precipitation, agglutination, neutralization, and complement fixation. In 12.4, there's only one autoimmune disorder you have to study, that is SLE. And the good thing about focusing in 12.2 is that by learning about B cells and T cells as well as antigens and antibody, you sort of also prepare for 12.1. So let's move on to discuss the questions that were tested from these two subtopics. The reason why we go through past year questions is not because we expect these questions to come out exactly, but so that we can familiarize with how the sentences are arranged when in actuality they are testing the same concepts. We'll start with the simplest question, the 1-mark question in 2013 repeat, that is definition. Biology definition questions are very common, whereby every concept you study, it is best you know the definition. However, it doesn't amount to many marks, it's maximum 2 marks. So your bigger concern will be the larger mark questions. So we look at the next question in 2013. Okay, here they ask you roles of lymphocytes. And you see a similar question was tested in 2019 repeat, where they ask you roles of B and T cells. Okay, only difference here it's 8 marks, here is 6 marks. Okay, but the answer is going to be exactly the same. The only reason why in 2013 they gave you 8 marks is because they did not identify what are lymphocytes. So if you are able to tell that lymphocytes are B cells and T cells, you get the extra 2 marks. So the answer technique, of course, will be to produce 2 paragraphs. 1 paragraph for a B cell and 1 paragraph for T cells. Okay, and since it's 6 marks, each cell will be allocated 3 marks. So to play safe, you write about 4 points per cell. Okay, but when studying, it's not only the roles of the cells you should know, you should also recognize their structure. So let me give you a little bit of theory here. In a T cell, on its surface, it has proteins called CD receptors. The important T cells are the alpha T, which have CD4 receptors, and cytotoxic T, which have CD8 receptors. B cells are completely different. Their surface proteins are called MHC, class 2 protein, and they also have immunoglobulins on their surface called IgD. Now, their function is completely different in that the B cells are involved in the humoral response. In humoral response, the B cells will produce antibodies that will bind to the surface proteins of the pathogen. The surface proteins of the pathogens are called epitopes. So when the antigen-antibody complexes form, some chemical reactions occur to destroy the pathogen. Okay, so this is humoral response. The T cells are involved in the cell-mediated response, where the T cells will proliferate and differentiate into cytotoxic T cells that will help to destroy infected body cells. Infected body cells are body cells that have already had the pathogen enter inside them. So body cells also have surface proteins. Their surface proteins are also MHC, just like B cells, but these are MHC class 1 proteins. Okay, so this is a brief introduction to the structure and function of the lymphocytes. And you can see the roles of these cells have been tested in 2013 repeat and 2019 repeat. Okay, let's see another pattern of question. Okay, this time you look at the 2018 repeat. They ask you to describe the action of the immune response against cells infected by viruses. Okay. Then we look at the 2019 repeat. They ask you to explain the production of antibody against bacterial infection in humans. Okay, sounds very similar. We continue with 2021. They ask you to describe the response of the body immune system towards pathogens that activate the B cells. And last year, they ask you to describe mechanism of immune response when a person's body is infected by bacteria. Okay, so at first glance, when you look at the question, it seems as if they are repeating themselves. The only difference being the microorganism involved. So like in 2018 repeat, they talk about a virus. 2019 repeat, they talk about a bacteria. 2021, they talk about a pathogen. 2022, they go back to a bacteria. But actually, regardless of whether it is a virus, a bacteria, or a pathogen, these microorganisms will either involve a cell-mediated immune response or a humoral immune response depending on its location. If it is circulating in the body, then this will stimulate the humoral immune response. If it has entered a body cell, then it will stimulate the cell-mediated immune response. So if we re-look at this question, you see here, for 2018 repeat, they say that the cell is infected, meaning that 2018's answer scheme will involve the cell-mediated immune response. Okay, I just put CM lah, cell-mediated. Now, 2019's repeat, they tell you, explain the production of antibodies. So the cell that produces the antibody is going to be the B cell. So this is involving the humoral immune response. So you see, 2018's question tested cell-mediated, 2019's question tested humoral. Let's look at the 2021 question. 2021 question is talking about the immune response involving B cells. So B cells are the ones that produce antibodies. So once again, this is humoral. So you can see the answer for 2019 repeat and the answer for 2021 is going to actually be the same. Only difference in the explanation, you have to use the word bacteria for 2019, but use the word pathogen in 2021. Okay. Then we look at 2022, last year's paper. Here, they say the body is infected. So if a body is infected, this is different from a cell is infected. So in 2022, you have to write both cell-mediated as well as humoral. So this is how you analyze how in-depth you have to write, or what are the choices you have to consider when answering. Let's look at one more question, that is the 2016 repeat. Okay, it's a little story where they are telling us that Sally was stricken by a disease, and after a month she was reinfected by the same disease. Okay, then they ask you to explain how the immune system responds. So now, if you focus on how the immune system responds, then you may be thinking of answering just like how you answered in 2022. But the keyword to note here is the word reinfected. So when you're talking about reinfected, we have to mention memory cells. So when you talk about memory cells, you have to mention the B memory cells and the T memory cells. You don't have to explain from the macrophage. So of course, 2016's answer is going to be completely different from all the others. Okay. So now, considering that 2022 was already an essay question, I would consider that maybe this year, if 12.2 is to be tested, it would be more of a structure question. So let's have a look at the examples of past year structure questions. This is the structure question from 2013. Okay, and the words here in red are actually the comments given by the MPM related to the kind of errors students were doing. Okay, so we look here, they've already given interaction between antibodies and antigens. So naturally, when it comes to antibody antigen, this is going to be the humoral immune response, right? So they ask you here to name structure X and state its function. So if you see, you recognize these to be antibodies. We know that they've mentioned here already, right? Antibody and antigen. So the Y shape is antibody. So this circle has to be antigen. And as I mentioned, antigens also on their surface, they have these proteins, but we call them as epitopes. So MPM states that many students do not know how to spell. Instead of saying epitope, they have been writing it as epitote. So please make sure for all scientific terms, memorize your spellings correctly. Okay, then in B, they ask you to name the cell that produces Y. So we know these Y are antibodies. They didn't ask us to identify what is Y, okay, but Y is antibody. So we need to tell which cell produces these antibodies. So out of habit, okay, we may say that it is B cell, okay, because they said name the cell and it's 1 mark. So naturally, we're going to think that you're going to answer it as B cells. Okay, but at STPM level, they want the answer to be more in detail. Okay, so some students, of course, they move on to say plasma B cells. Okay, but like I said, MPM requires very in-depth answer whereby you have to tell the cell that produces Y are actually two types of B cells, that are the plasma B cells as well as the memory B cells. Because the memory B cells are the ones that will produce antibodies very fast upon reinfection. Okay, so this is going to be quite a challenging mistake because normally students will not think beyond plasma B. Then for question C, they ask you to state two functions of the antigen-antibody complex. Okay, so basically we know in this case where you can see that one antibody is like pairing with two antigens. So this is clearly an agglutination process. Okay, so normally when you are writing, you will mention agglutination occurs so that the macrophage can then digest. okay, or destroy the antigen. So MPM again comments that sometimes students mention that the agglutination will occur so that the macrophage can engulf the complex. Okay, but they don't want you to stop at engulf, because the macrophage not only engulf means swallow, not only swallows the antigens, but it will also digest it. So that's why you must make sure when you write macrophage, you mention engulf and digest. Okay, so these corrections that we are talking about is not only for this question. Okay, learn to use it in any context. Meaning that if you are going to write about antibodies, make sure you mention plasma B cells and memory B cells. If you are going to mention anything about macrophage, make sure you mention engulf and digest. Okay, then in D, what type of immune response? So as I mentioned, if it's antigen antibody, you know it's going to be the humoral immune response. And here they ask you the role of the helper T in the immune response mentioned. So, you know, helper T, its function will be to synthesize interleukin 2. Okay, only again a reminder, students sometimes go and write interleukin 2 using this symbol of 2. So please remember, interleukin 1 and interleukin 2 are written using the normal mathematical numbers. So this was 2013. Let's look at the next structure question. Here you see in 2018, 2013 already tested humoral immune response. So 2018 this time is testing another immune response. Here you can see there's no picture of antibodies, but there is an infected cell. So this tells us this is going to be the cell-mediated immune response. So that's the same thing they are testing here. And then you have to identify. So when you are studying for this chapter, make sure you know how to label all the cells in a diagram. So here they've labeled macrophage for us. Okay, so when a macrophage has ingested and digested a pathogen, remember the macrophage will become an APC, antigen presenting cell. So because of that, the helper T, okay, so the helper T will come to help. So when the helper T forms a complex with the APC, remember the macrophage will begin to synthesize interleukin 1, which will then stimulate the helper T to synthesize interleukin 2. And this interleukin 2 then will cause helper T to form clones that will differentiate into a variety of T cells, which can be cytotoxic T, suppressor T, helper T, and memory T. So once you understand this cell-mediated immune response process, then identifying P, X, and Y is simple. Okay, only thing as usual, as mentioned earlier also, students kept writing interleukin number 1 here using this symbol, or interleukin 2 using this symbol. So please, in 2013, MPM already asked students to be alert, but in 2018, students are still making this mistake. So I hope in 2022, you will not repeat this mistake. Okay, then when they ask you to describe the process at stage 1. Okay, so at stage 1 here, if this is 2 marks, so you cannot just mention that it will synthesize interleukin 1. You have to talk about how it becomes an APC. So the same thing here, you see, macrophage, engulf, and digest. The same correction of 2013 is being mentioned in 2018. Then we move on to D. What is the effect of Y on Q? Okay, so here, even when I mentioned just now, I failed to use the keyword that they expect. I only mentioned that cloning occurs, but the keyword they want is that interleukin 2 will activate proliferation of T cells or stimulate proliferation of T cells. So especially when it comes to structure questions, use of correct keywords is important, right? Finally, they ask you to identify the substance released by R. So remember, if it is a cytotoxic T cell, it will synthesize perforin that will perforate the infected cell, causing it to lyse. So spelling problem again, perforin is spelled without an E. So you see the two structure questions that came out, one tested humoral immune response, one tested cell-mediated response. But since cell-mediated response was more recent, I would anticipate if a structure question were to come out this year, it should be on humoral response, or maybe it could be both humoral and cell-mediated, or maybe they may test on the memory part. So please do read up these parts of the topic. The next subtopic, 12.4, immune disorder, is very easy to prepare because, as I said, there's only one disease. Make sure you get the spelling of systemic lupus erythematosus correct. And you can see basically when you are studying this disease, you just need to know what is the disease, why a person gets that disease, and how you can prevent outburst of this disease. So I hope with the suggestions that I've talked about and discussed, you are more clear on how to approach your revision. However, I will be adding another video soon on the cell-mediated and humoral response. So until then, happy studying. Bye-bye.